that we are in the middle of cellular respiration, let's talk about the phases that occur in the presence of oxygen. So we finish glycolysis and we have 2 3-carbon molecules. The next thing that's going to happen is a linking reaction where we're going to take that pyruvate and we're going to change it to a compound known as acetyl-CoA. This is going to release carbon dioxide and it's going to give us another electron carrier. So each of the pyruvates is going to give us an acetyl-CoA. But notice acetyl-CoA is only a 2-carbon molecule. And so that's how carbon dioxide gets released. And it also gives us an electron carrier. So this acetyl-CoA will then go into the into the second multicellular process, or the third step overall, known as the Krebs cycle or the citric acid cycle. Here, we're going to be making more ATP as well as more high energy electron carriers. Both the linking reaction and the citric acid cycle occur in the solution part of the mitochondria, in the mitochondrial matrix. So when you look at this picture on the right, you can see the pyruvate becoming acetyl-CoA, and now it's entering this cycle of reactions known as the citric acid cycle. So we have the 2-carbon acetyl-CoA, and the CoA is going to break off from it, and that 2-carbon molecule is going to bind to the 4-carbon molecule. And as you can imagine, what's that going to require? Anyway, that forms citric acid, from which we are going to release a carbon to go down to a 5-carbon molecule and also create electron carriers. We're going to release another carbon as carbon dioxide to go to a 4-carbon molecule, release another electron carrier, as well as create some ATP. Now, this 4-carbon compound will make 2 different electron carriers and will stay as a different 4-carbon compound, and will join up with more acetyl-CoA and go around the circle again. So each cycle produces 1 ATP, 3 NADHs, 1 FADH2, and 2 carbon dioxides. But remember, we got 2 pyruvates from each glucose molecule, so there are 2 citric acid cycles for each glucose molecule. So overall, for each glucose molecule, we're going to end up with 2 ATPs and 6 NADHs and 2 FADH2s and 4 carbon dioxides. And yes, did you pay attention to whether we were doing hydrolysis or dehydration synthesis? This is going to take water to do these reactions. Okay. So you may be asking yourself, what are NADH and FADH2? As I said, they are electron carriers, high electron carriers. So kind of think of them as sort of the same but different. So sort of like Uber and Lyft. Okay. They are basically carrying these energies as electrons to the last process, this third multi-step process that's going to occur, that electron transfer chain. There's going to be another difference in that when they get to the electron transfer chain, where they drop their contents is going to be a different. So they have different places where they can pull up and drop off their passengers. Okay. NADH can drop off at the beginning, so it's going to be yielding 3 ATPs for every NADH, whereas FADH2 can't start at the beginning. No, it's prohibited. So it can only start partway through the process. So each FADH2 will only yield 2 ATPs. But we will see that right now as we go through the steps of the electron transport chain, because this is where ATP formation occurs. So when you look at your mitochondria, it has this outer membrane and an inner membrane that is folded, and these infoldings are called the mitochondrial cristae. And on the cristae, we've got these proteins, which are represented as these purple globs right here. And so they go across the cristae, okay? And so as it comes along here, you can see NADH going at the top of this electron transport chain. So basically, we've got this whole row of proteins, this electron transport chain. Just think of them like steps on a stair. There's a whole row of them. And NADH gets to start at the top step. And when it comes along, it releases its hydrogen ion right here into this into the space, right here in this purple area, into the space between the two membranes. And the hydrogen ions are going to be floating down there, okay? And the electrons are going to be going down the steps, okay? And they're going to create a concentration gradient. Now, when FADH2 comes along, it can't do the top step, but it's going to go partway down. And so just think of it like there's, like, think of it like a bunch of fraternity brothers passing stuff down and down and down and down, going down the steps, okay? And as these electrons are traveling down the steps, when they get to the bottom, what they're going to see down at the bottom is they are going to see oxygen as the final electron acceptor. You don't have to know any of the other electron acceptors, but oxygen is down there, okay? So oxygen accepts the electron. Meanwhile, these hydrogens have been cruising along the space between, and then they find a channel and they are going to come back in, and oxygen's going to pick up the hydrogen. And what does it form? Our friend water. Okay, so here's where we get our waste product of water right there. In addition, the kinetic energy of this hydrogen is going to give us the power to form a bond between ADP and a phosphate to make ATP, to convert it. So here is where ATP is being made. Okay, so once again, electron transport chain going down, 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 NADH, FADH2 drop their electrons, they go down the energy chain. That's going to bind with and each one's going to bind with the oxygen. Their hydrogens are going to go and combine with the oxygen to form water. And this controlled release, the energy from the hydrogens, is going to enable us to synthesize ATP. So take this moment, review these three multi-step processes in aerobic cellular respiration, and don't forget to put in that fourth process, that linking reaction. And so stop it and then return back, because I'm not quite finished. So where did we make our energy? We talked about energy being made in glycolysis, energy being made in the Krebs cycle, and energy being made in the electron transport chain. The energy in the electron transport chain was being made from the electrons being carried from the NADH and the FADH2 from the glycolysis and the Krebs cycle. So in a perfect world, we would have a net yield of 36 ATP for one glucose molecule. But remember at the beginning, I told you all of our food macromolecules enter cellular respiration. So sugars, if it's glucose, we've talked glucose, we're going to end up with 36 ATP. If it's fat, remember our lipids are broken down into glycerol and fatty acids. So some can enter through glycolysis, but some can only enter here at the beginning of the citric acid cycle. So how many less ATP could they make? Some amino acids, like for instance, pyruvate, will at the end of as pyruvate. It's not going to be making 36 ATP. Some amino acids will be starting at the linking reaction. Some amino acids will be starting at various places in the citric acid cycles. So you can see with proteins, we have a totally variable number of ATPs we can make from each protein. So as you can see, when we talk about what is the ideal source of energy, this is why athletes do carbohydrate loading. Those simple sugars that easily become glucose monomers are going to give the maximal amount of ATP as opposed to those proteins and those fats. So once again, take a moment. I like this picture to the right because it's very simple, shows you the 4 steps, shows you how much energy is being made from each one. A net of 2 ATPs from glycolysis, a net of 2 ATPs directly from the Krebs cycles. But this picture on the left is showing you that electrons are being carried from NADH from glycolysis, which is occurring in the cell, and electrons being carried from NADH and FADH2 from the Krebs cycle occurring in the mitochondrial matrix, going to the transport chain and in the mitochondrial cristae, which is going to give us 32 ATP. And that's it for what happens with oxygen. Please return once you understand this and teach somebody for our last video, which is what happens in the absence of oxygen. So I'll see you soon.